Cellexus Limited is a UK-based company founded in 2008 and now based in Cambridgeshire. Since then, the company has brought to market a range of innovative, next-generation, single-use, disposable bioreactors which use a novel airlift technology designed by Cellexus to simplify and improve fermentation and cell culture bioprocesses in biotechnology research, scale-up, production and contract manufacturing. The company launched the Cellmaker Regular for cell culture of bacteria, yeast, bacteriophage and algae in 2008. It followed this with the launch of the Cellmaker Plus in 2011, which included pH and DO sensors with capability of culturing most cell types, including insect lines. The systems incorporate the innovative single-use disposable bioreactor technology from Cellexus, as well as the unique airlift technology which provides optimal aeration and mixing, as well as precise temperature and dissolved oxygen control for improved cell culture. The company has made further significant design and operational improvements and is now relaunching the Cellmaker Regular and the Cellmaker Plus with enhancements to provide users even easier to use control, increased flexibility and improved robustness. The systems are easy to use with improved performance to achieve optimal yields of soluble protein using the single-use cell culture bioreactor Cellmaker bags. The Cellmaker systems are essentially composed of two main units, the Cellmaker Controller and the Cellmaker Enclosure. The Cellmaker Controller, seen here on the left, is the brains of the system. It incorporates an air pump and handles temperature regulation. On the Cellmaker Plus system, pH and dissolved oxygen are also incorporated. It also enables a process measurement, data acquisition and provides a power source. The controller enables user-defined parameters to be defined, such as temperature, and gas mixing via a touchscreen monitor which can be positioned on top of the controller to reduce bench space if required. Control of dissolved oxygen is crucial for optimal cell concentration and viability, and the unit comes with pH and oxygen sensor readers to enable these parameters to be measured. Automated feedback of data enables pH control as well as regulation of adjustment of air, oxygen and carbon dioxide. The Cellmaker Enclosure is essentially a housing to provide support and connections to disposable Cellmaker bag. The Cellmaker bag is a vessel in which the cells are grown. The Cellmaker Enclosure incorporates a Peltier-driven thermal plate and temperature sensor to enable the cell culture temperature to be closely monitored and controlled. The enclosure comes in 8 litre and 50 litre sizes for use with the 8 litre and 50 litre Cellmaker bags respectively. The 8 litre enclosure can operate from 1 to 8 litres and the 50 litre enclosure can operate from 10 to 50 litres to provide scale flexibility. The enclosure also comes with transparent panels to enable visualization of the cell culture process and to enhance performance in light-dependent applications. The Cellmaker bag is a unique patented design employing a state-of-the-art airlift technology which is simple, scalable and inexpensive to use. The Cellmaker bag is designed to aerate the cell culture from the base and features an integral white sparge tube which runs the full width of the base of the Cellmaker bag. The sparge tube delivers gas to the Cellmaker bag, enabling efficient aeration and mixing of the culture media by the unique aeration principle. The relatively large surface area of the Cellmaker bag, with respect to its volume, allows gentle gas exchange for cultures that do not require vigorous aeration. The Cellmaker bag is supplied gamma irradiated to ensure sterile conditions. The system is usually set up in the laboratory with the gas delivery components towards the left and the liquid delivery components towards the right. To minimise bench space, the touchscreen monitor can be placed on top of the controller, as shown earlier. On removing the Cellmaker bag from the packaging, a number of air filters are fitted to enable maintenance of the sterile integrity of the Cellmaker bag, which is essentially the culture vessel. The filters can be attached on the laboratory bench. Users can use 70% alcohol to maintain sterility as the attachments are made, but should users require additional sterility to avoid yeast or viral contamination, attachment to the filters may be done in a laminar flow hood as illustrated here. For further additional security, users can also attach cable ties to the tubing attachments, which is also illustrated here. A highly efficient condenser system is incorporated for the removal of moisture from the exhaust line via a microbiological filter to prevent contamination. Optionally, gamma irradiated single-use condenser liners are also available to enable ease of use as well as fast setup and turnaround times. The Cellmaker bag is easy to load into the Cellmaker enclosure, simply sliding the bag into the enclosure unit and being retained in place by lockable side panels. The final connections between the Cellmaker enclosure and the Cellmaker bag are made. In the case of the Cellmaker Plus, the Cellmaker Plus bags come with integral single-use pH and dissolved oxygen sensors, and connections are therefore made between the pH and DO sensor ports and the respective pH and DO sensors. Fibre optic probes permit reading from the probe patches attached to the ports at the back of the Cellmaker bag, and signal is delivered to a reader device housed in the controller, which converts the signal and permits control via a feedback loop. The Cellmaker bag is first filled with air to prevent creasing of the Cellmaker bag during installation and facilitates the best fit of the Cellmaker bag into the Cellmaker enclosure. For convenience, a peristaltic pump is used to fill the Cellmaker bag with the appropriate media. Pre-sterilised media can be pumped directly into the Cellmaker bag, or non-sterile media can be pumped into the bag via a sterile media filter. If the media filter has been pre-autoclaved, it may be necessary to pre-wet the filter with media before pumping to prevent filter blockage. Once the Cellmaker bag is filled, the media line is clamped off. The gas supply is delivered to the Cellmaker enclosure via the hose on the right of the enclosure. Gas enters the Cellmaker bag via the sparge tube. The cable on the rear of the enclosure delivers a low voltage power supply and allows communication between the Cellmaker controller and the Cellmaker enclosure. The reciprocal connections exist on the Cellmaker controller unit. The Cellmaker controller is operated by the touch screen. To ensure the temperature probe is stabilised, and in the case of the Cellmaker Plus, that the sensors are suitably hydrated, a stabilisation step of approximately 30 minutes is performed. A dissolved oxygen calibration cycle, which takes approximately 20 minutes, is then conducted. The temperature is now set via the touchscreen and adjusted by the scroll keys. The temperature is controlled by Peltier plates in contact with the Cellmaker bag and monitored and controlled throughout the culture process using an integrated temperature probe. The system has capacity to cool as well as heat the culture media. Ability to lower culture temperature may be advantageous for expression of toxic or poorly soluble protein in E. coli. The flow of air and oxygen is controlled by setting user-defined values via the touch screen. Up to 10 litres a minute of air can be delivered to the Cellmaker bag and up to 4 litres of oxygen. On the Cellmaker Plus, oxygen flow is automatically controlled by monitoring of the DO using the integrated DO sensor in the Cellmaker Plus bag, such that if oxygen levels fall below the user-defined levels, oxygen will be added to ensure that that level is at least maintained. Air supply to the headspace at the top of the bag helps to increase gas exchange and reduce foaming. The vent regulates the flow of the exhaust gases and dictates the overall pressure in the Cellmaker bag. 3 inlet ports can be found at the top of the Cellmaker bags, which enable addition of acid and base to control pH, as well addition of anti-foaming agent and glucose should that be required. On the Cellmaker Plus, a pH calibration step is performed, and acid and base addition is automatically controlled via monitoring the pH using the integrated pH sensor in the Cellmaker Plus bag and automatically delivering acid and base via a peristaltic pump as required. The inoculant is added via a sample port at the right hand side of the Cellmaker bag. The sample ports can also be used to enable samples to be taken or to allow addition of reagents to the media during the culture process. In summary, the key benefits of the Cellmaker systems include temperature control in the 18 to 40 degree range, allowing for low temperature induction of protein expression in E. coli. Temperatures are maintained within plus and minus 0.1 degrees centigrade. Gas flow control, a key feature which enables optimal growth of oxygen-hungry bacteria and yeast. Single-use disposable cell culture bags which are uniquely designed to provide the most efficient aeration and mixing by the airlift principle. Inclusion of pH and DO sensor technology on the Cellmaker Plus system, which enables automated control of pH and dissolved oxygen. Inclusion of a pH sensor to enable addition of acid or base to precisely control pH. The Cellmaker systems are manufactured in the UK at the Cellexus facilities in Cambridgeshire and sold worldwide.